basic sources of fuel that we use for energy, we will discuss how those fuel sources are used in metabolism. So the first set of topics and outcomes are as follows. And yes, I said the first set. Okay, so we're going to focus on ATP and an energy system called the ATP-PCr system. So we need to produce and release energy in order for our body to complete its normal function. Energy is released when ATP is broken down into ADP. This happens when a chemical bond is broken, which releases stored energy. Some of this released energy is able to be harnessed in order for our body to be able to function. Interestingly, our body stores very little energy. If we stored all of the ATP that we would need for a day, it would be very inefficient, as it would significantly add to our weight, which would in turn increase our energy requirement. Consequently, we need to have the ability to produce ATP when we need energy. Now, there are three primary systems that are used to create ATP. One of these is called the ATP-PCr system, or the ATP phosphocreatine system. While our cells do not store very much ATP, they do store a compound called phosphocreatine. The ATP-PCr system takes phosphocreatine and, with the help of an enzyme called creatine kinase, phosphocreatine is broken down into phosphate and creatine. This phosphate that has been released from creatine can be added to an ADP molecule, which then forms ATP. This ATP can then be broken down to release energy for use. Now, while we are constantly using the ATP-PCr system, it is the primary energy source at the onset of exercise. After 10 to 15 seconds, our phosphocreatine stores are depleted, and we are unable to generate very much ATP with this system going forward. Now, in the case of the ATP-PCr system, since there is only one enzyme, it shouldn't be a surprise that creatine kinase is that rate-limiting enzyme. As just discussed, this enzyme is important in creating ATP from phosphocreatine, because creatine kinase breaks down phosphocreatine. Consequently, the activity of this enzyme is closely tied to ATP concentrations. When ATP concentrations are high, the activity of creatine kinase is low, and vice versa. So this is a great example of a negative feedback loop. High ATP, we've reduced the enzyme activity of creatine kinase, which means we're not going to form as much ATP. So, this will be one of the few lectures where I have a second learning topic and outcome slide. This is only because if I tried to fit all of the topics and outcomes on a single slide, it would be hard to read. And so the last half of this lecture is going to focus on glycolysis. At its simplest, glycolysis is a 10-step process that takes glucose and creates several things, including ATP. However, instead of focusing on the individual 10 steps, I want to break it into two halves. The first half, or phase, is often referred to as the investment phase. A glucose molecule undergoes 3 steps that results in the formation of 2 identical molecules. However, it costs 2 ATP to create these molecules. In other words, we're trying to use glucose to make energy, but at the beginning we've actually spent energy. So we're down 2 ATP. The last 7 steps are referred to as the generation phase. A lot of things go on, but ultimately a total of 4 ATP are created, which means a single molecule of glucose can be used to create 2 ATP. Remember, we spent 2 ATP in the first part, we gained 4 ATP in the second part, so 4 minus 2 is 2. It is also important to note that 2 molecules of NADH are formed during this part of glycolysis. And NADH will be used during aerobic metabolism, specifically during the electron transport chain. Additionally, it's important to know glycolysis does not require any oxygen. So glycolysis occurs in the cytosol of the cell. And although glycolysis doesn't require oxygen, the availability of oxygen determines what is ultimately produced at the end of glycolysis. If oxygen is available, 2 molecules of pyruvate are formed. However, if oxygen is unavailable, 2 molecules of lactate will be formed and not lactic acid. Okay? Glycolysis is the primary energy system once the ATP-PCr system is depleted, and continues to be the primary energy system for all-out events that last anywhere from 2 to 3 minutes. Phosphofructokinase is an enzyme that is in fact the rate-limiting enzyme for glycolysis. Phosphofructokinase is the enzyme that allows that third step of glycolysis to occur. Okay, and this is a three-dimensional spinning model of it, right? All right, so just like creatine kinase, phosphofructokinase is sensitive to ATP concentrations. As ATP concentrations increase, that means we've got a lot of available ATP, which means we don't need to really go through glycolysis, so phosphofructokinase activity decreases. However, if we have low ATP concentrations, the activity level of phosphofructokinase is increased. So again, an example of a negative feedback loop. The final thing I want to mention about glycolysis is that glycogen, remember that storage form of glucose, can also directly enter into the glycolytic pathway. When this happens, the investment phase only requires 1 ATP, so glycogen results in the formation of 3. So glycolysis, when we use glycogen, results in the formation of 3 ATP instead of only 2 ATP. So after this lecture, are you able to answer these questions about the ATP-PCr and glycolytic systems? Can you explain how the ATP-PCr system works? Can you describe what occurs during the investment phase of glycolysis, as well as what happens during the payoff phase of glycolysis? And can you explain how oxygen affects glycolysis?